晚上好，欢迎大家来到 IBA 和达科维生物共同主办的达优学堂直播间。很高兴今晚和大家一起学习在线课程。本次在线课程请到 IBA Life Sciences 的产品经理 Dr. Alexandra Notlona 来给大家分享 MHC 多聚体在抗原特异性 T 细胞分选与分析中的作用。Dr. Notlona 将从 MHC 多聚体及其在抗原特异性 T 细胞研究中的作用、可逆多聚体 MHC Streptamer 在分选和 TCR 分析的应用，还有关寻找适合临床治疗中功能性 TCR 的实验设计思路这3个方面进行介绍。在听课的过程中，各位新进来的小伙伴，如有直播软件问题，可以在讨论区留言，这里有小助手帮大家解决哦。如有 MHC 多聚体及抗原特异性 T 细胞分选和分析相关的专业问题，也欢迎大家在讨论区提问。IBA 的技术专家将在讲解结束后移步到微信交流群，统一解答您的提问。课程结束之后呢，我们的微信群里将进行有奖竞答活动，问题的答案都在讲师的课件里，看谁是最爱学习的人。好，接下来的时间有请 Dr. Notlona。
Hello everyone, welcome to this Dakewe webinar. My name is Alexandra Nordlohne and I'm a product manager at IBA Life Sciences. In this webinar, I will talk about how MHC multimers can be utilized for T-cell receptor isolation. This talk focuses on CD8 positive T cells, which are also called cytotoxic T lymphocytes. These cells are part of the adaptive immune system, and their main function is the recognition and elimination of faulty cells, so for example, cells that are infected with the virus or have mutations that can potentially lead to the development of cancer. In autoimmune diseases, this recognition goes wrong, and T cells actually recognize and kill healthy cells. However, this webinar is about the beneficial function of T cells, as this feature can be used for immunotherapeutic approaches. Researching T cells and their receptors helps us to understand immune responses and develop treatments, for example, for cancer. Isolating T cells with T cell receptors, for example, specific for a cancer antigen, and subsequently producing high numbers of T cells that express this TCR, which then in the end can be transferred back into the patient, is one possible treatment option. How exactly this works and which steps are involved, you will learn in this webinar. But let's start with some general information about CD8 positive T cells. An adult human has around 40 billion cells of this T cell subtype, and these cells recognize antigenic peptides via their T cell receptor. The T cell receptor is a heterodimeric receptor consisting of an alpha and a beta chain, which undergo somatic recombination and nucleotide insertion during maturation. This way, we have around 1 to 10 million different alpha-beta T cell clones, and therefore around 4,000 to 40,000 cells per clone. In total, all these T cell clones recognize a broad range of different antigens and thereby provide protection from infections or cancer. The binding to and recognition of antigens is mediated via major histocompatibility complexes, short MHCs. They are subdivided in MHC class 1 and MHC class 2 complexes, and CD8 positive T cells specifically bind to MHC1. MHC1 molecules are expressed on the surface of our nucleated cells, where they present short peptides from endogenous proteins. This means that both own peptides, but also, for example, peptides derived from a virus that replicates inside a cell, are presented. The job of T cells is to identify these cells that present non-self antigenic peptides and kill them, which is why they are also called cytotoxic T cells. In contrast to antibodies, TCRs do not undergo somatic hypermutation and affinity maturation during the course of an immune response. This means that the affinity between a TCR and a peptide-loaded MHC molecule does not change. This affinity is actually quite weak, as the dissociation constant generally ranges between 100 and 1 micromolar, which is up to 10,000 times lower than that of some very high affinity antibodies. I put the formula for the dissociation constant on the slide, which is calculated by dividing the off rate K off by the on rate K on. The off rate is given in seconds and independent of a concentration, whereas the on rate is given in mole per second and is actually dependent on the concentration. A slower off rate, which means that the MHC and the TCR bind for a longer time, correlates with improved T-cell functionality, for example, the killing capacity. That is why the dissociation rate is a highly stable biomarker for T-cell potency. The off rate will come back later in this presentation, as it is important for selecting the best functioning T-cell receptors. The weak affinity of the TCR to MHCs proved to be a challenge in immunology research, as staining, for example, a T-cell population using the corresponding labeled MHC is not as easy as using a labeled antibody. The MHC binding to a single TCR is so weak that it is not possible to directly use it, for example, for fluorescent staining. Therefore, the former standard method for analyzing antigen-specific T-cells was limiting dilution analysis. This method, however, significantly underestimates the actual frequency of antigen-specific cells, as cells without proliferative potential are not detected. In addition, this approach is very time-consuming, as it can take up to 5 to 7 days to get a final result. To overcome this problem of fast dissociation of MHCs from TCRs and actually use these molecules for direct staining of T-cells, Altman and colleagues generated the first MHC multimers in 1996. They multimerized biotinylated MHC complexes on fluorescently conjugated avidin. Nowadays, usually strep avidin is used instead of avidin. This way, several MHC molecules bind to the cell at the same time, which increases the stability of the staining complex. The so-called tetramers allowed the staining of T-cells independent of their functional status, and this approach actually revolutionized the field of immunology research. This slide shows the first successful staining of antigen-specific T-cells using tetramers. Altman and colleagues stained the A2 pole-specific CTL clone 20, so a clone specific for HIV-derived pole peptide, with tetramers specific for A2 pole, or with tetramers against A2 GAG, so a different HIV-derived antigen. They showed that only the specific tetramer could stain these cells, which is actually the proof of principle for using tetramers for antigen-specific T-cell analysis. They also showed that these tetramers can be used for sorting cells. They sorted an influenza matrix peptide cell line into positive and negative fractions and demonstrated that the cells are still functional as the positive fraction efficiently lysed matrix protein pulse target cells. Since the first generation of MHC tetramers in 1996, several other methods for multimerization were developed throughout the years. The underlying principle of pentamers is that 5 MHC molecules are multimerized via a coiled domain and connected to 5 fluorophores. In the streptamer technology that was developed in 2002, MHC molecules are fused to a streptag and multimerized on streptavidin derivative called streptectin. And in dextramers, MHC molecules are multimerized on a dextramer backbone. The special feature of streptamers is that they were actually the first reversible multimers that were developed. The reason why a reversible staining option was explored is the fact that irreversible binding of multimers to T cells can potentially affect their function. For example, in 1999, Whelan and colleagues observed that as soon as the isolated cells are cultured at 37 degrees, the multimers get internalized into the cells. 2 years later, another study showed that T cells undergo apoptosis if cultured with irreversible multimers. This demonstrates that there was a need for reversing the binding between multimers and T cells to minimize the effect of staining procedures on T cell function. The solution to this problem was the development of MHC1 streptamers in 2002 by Knabel and colleagues. They used the streptavidin variant streptectin as backbone and multimerized MHC molecules via a streptag. This resulted in a stable binding to cells. However, addition of biotin could actually reverse this binding and cause the dissociation of staining reagents from the cells. This MHC1 streptamer approach is based on our proprietary StrepTag technology. We produce MHC molecules with antigenic peptides that are fused to a twin StrepTag, which are called MHC1 streps. The twin StrepTag is an amino acid sequence that binds to the biotin binding pockets of streptactin. By combining MHC strep with streptactin, you get multimerized molecules called MHC1 streptamers. Because biotin has a much higher affinity to streptactin than the twin StrepTag, it causes the release of MHC1 streps from streptactin. The resulting monomers don't stably bind to cells and also dissociate from the surface. In the study from 2016, streptamers were compared to pentamers, and it was demonstrated that streptamers can still give highly accurate results, even if the frequency of antigen-specific cells is very low. The authors diluted PBMCs of HLA, O1, O2, CMV seropositive healthy donors with PBMCs of negative donors. Even at a 64-fold dilution, MHC streptamers gave a value that was very close to the theoretical one. The authors also tested the precision of streptamers and pentamers, and this was actually very similar. The key advantages of MHC streptamers, in addition to precise and accurate measurements of T-cell populations, is that in the end you get completely label-free and fully functional cells due to the reversible labeling reagents. At the same time, the purity and viability of cells is very good. In the next part of this webinar, you will learn one possibility of how to use MHC streptamers to isolate T-cell receptors suitable for clinical approaches or to answer basic research questions. I will talk about all steps that are needed in this process, starting with the isolation of T cells, followed by identifying TCR sequences, which then, in the next step, after cloning them into target cells, need to be functionally characterized to finally provide a possible clinical treatment option for patients. But keep in mind that there are several different ways to reach this goal, and I'm presenting only one option today in more detail. I will start with briefly introducing a direct method to isolate the TCR sequences of multimer-stained antigen-specific cells, which was developed at the Technical University of Munich. They called it T-cell receptor single-cell analysis method, short TCR-scan, and in this approach, single T-cells are sorted directly onto PCR slides. Afterwards, direct gene-specific reverse transcription was performed, followed by different PCR steps that are described in detail in this publication. They optimized all steps in this procedure, for example, by using an oligo-detailing to generate an artificial priming site, by using gene-specific primers for alpha and beta chains instead of standard oligo-DT primers, and by finding the optimal temperature for the reaction. They tested the approach by sorting T cells specific for a peptide from CMV using multimers. They analyzed 36 single cell samples from this donor, and the boxes in this picture highlight those samples that yielded full-length alpha and beta chains. Although this approach can be successful, it is actually quite difficult to perform as it needs a very precise cell sorting process, making the initial isolation very error-prone. This is especially challenging for very rare T-cell populations from antigen-unexperienced donors. In addition, there is only a very low amount of RNA present in a single cell, potentially reducing the success rate of PCR reactions. I will now introduce possible optimization steps for these challenges, such as pre-enrichment of the sample before sorting and single-cell culture after antigen-specific T-cell isolation. Now we're looking more detailed at step 1 of the whole process, which includes different experimental steps like pre-enriching the sample, staining the cells with multimers, sorting them, and optionally expanding them over a short term. But first let's start with the pre-enrichment step. To improve the complicated sorting process, we propose a step that already pre-selects the cells to reduce the amount of non-target cells in a sample. This can be done in an already antigen-specific way or by isolating T cells before the cell sorting process. Pre-enriching the sample before heading to the sorter can be crucial to reduce the sorting time, decrease staining background, and to increase efficacy of the sorting process in general. We propose the isolation of CD8 positive T cells as a first step if you're using MHC1 multimers for the isolation of cells. This way, the potential target cells are already enriched, but you don't make a too strict selection in the first step. Here we show a fast method using CD8 positive PubTax method with agarose columns for affinity chromatographic cell isolation, but of course, other ways of pre-enrichment are possible. The CD8 PubTax kit is also based on our StrepTag technology and has the advantage of being very fast, of having a high yield, as you do not lose 30 to 40% of your cells during PBMC generation, because you can actually isolate the cells directly from blood preparations such as Buffycoat, and as the isolation label is released from the surface during the isolation process, label-free and fully functional cells. After this pre-enrichment, we want to now look at the staining with MHC1 streptamers. As illustrated by Charisse and colleagues, different donors can have varying sizes of antigen-specific populations, and even with pre-enrichment, the percentages are very low. In general, virus-specific cells are more abundant than cancer-specific T cells. For some epitopes, the amount of naive CD8 positive precursor cells recognizing it can be as low as 80 to 1,200 cells. This makes it essential to have as many starting cells as possible to increase the chance of finding a fitting T cell receptor. After the pre-enrichment, the next step would be the sorting of antigen-specific T cells from that population. To illustrate the sorting, here you can see the gating of B7pp65 specific CD8 positive T cells by Effenberger and colleagues using MHC1 streptamers to sort for CMV-specific cells. They used MHC molecules with trin StrepTag in combination with streptactin APC to generate streptamers. This again highlights that although cells are pre-gated for CD8, the actual target population is only 1.9%. The sort was performed as a bulk, as single cells cannot be made visible in the fax machine. Using the MHC1 streptamers, they were able to sort a highly pure cell population only containing the target cells of interest. As mentioned before, sequencing a single cell can be tedious, error-prone, and might have a low efficacy. Therefore, we want to look at the second bottleneck, the low efficacy of single-cell T-cell receptor PCR steps due to the low amount of mRNA available in a single cell. Here we want to introduce a short single-cell culture after the isolation of cells, which makes the PCR way more efficient as the mRNA content is drastically increased. As a first step, the single cells should be sorted into 348 well plates, and each single cell should be expanded feeder-free, so without other cells in the culture. This aspect is important not to add additional, potentially interfering mRNA to the culture. The expansion needs 12 to 14 days, but one single cell can expand to significant populations, which dramatically increases the amount of mRNA, which makes the reverse transcription in the following steps way more efficient. The time invested here can reduce the time needed later on and increases the chance to get most out of a valuable donor material. But as mentioned before, this expansion is optional. As expanding single cells in culture can be tricky, we recommend using CD3/CD28 streptamers combined with 50 units per ml IL-2 in the cell culture medium. After a few days, single cell colonies should be visible under the microscope and be ready for further processing. The next step after successful isolation and expansion of T cells is the identification of T cell receptor sequences. In addition to the TCR scan method developed by Dössinger and colleagues that provides a method for single cells, there are several other methods for amplification of TCR sequences and generating TCR libraries. This picture shows examples of some other methods, including multiplex PCR, target enrichment, or rapid amplification of cDNA ends short-base PCR. As discussing the methods in detail is beyond the scope of this webinar, I recommend the cited papers mentioned here for further information. In this last step in the bench-to-bedside project, the isolated TCRs now have to be tested regarding their functionality. We only can give you some ideas how MHC multimers can help you to better understand the functionality of the isolated TCRs and to judge the biological significance of the TCRs and maybe even their clinical potential. We are fully aware that there's a multitude of other assays giving interesting data, and we also left out in vivo studies which can also provide you with some interesting data for TCR functionality. The first step in any of these assays is the successful re-expression of the TCR in a target cell. Always coming from an unedited donor cell, different methods are possible here, as for example shown in the study by Schober and colleagues in 2019. The most commonly used but also most artificial process is viral transduction. However, here you get the expression of both the physiological TCR and the transduced TCR, which can lead to suppression of the transgenic variant or to mispairing of the TCR chains. Therefore, a different approach is to use viral transduction, but to inhibit expression of the endogenous TCR. Viral transduction leads to a semi-random insertion of the TCR into the genome, which could be a concern in clinical applications. So more advanced methods use CRISPR-Cas9 to insert the TCR at the locus of the endogenous TCR to get a physiological regulated expression. For more details, you can look into this publication. After the TCR is re-expressed, MHC1 multimers can help here to evaluate specific TCR expression and thereby judge the transduction success. This way you can choose the system best suited for your experimental requirements. In this case, they stain T cells after re-expressing the TCR with different methods, and again after sorting and expanding antigen-specific T cells. MHC streptomers can also help to judge the functionality of TCRs independent from the current activation state, the transduction success, and other TCR-unrelated factors for T cells, as shown by, for example, Nauert and colleagues in 2013. The K-off rate assay is a way to measure the structural affinity of the TCR independent of before mentioned influences like surface expression and activation state. As I mentioned at the beginning of this webinar, the slower off rate correlates with T cell potency. In this assay, you stain your cells with MHC streptomers and then you measure the dissociation after disrupting the staining complexus biotin. Here it is important that the MHC monomers are diconjugated so that you can observe the dissociation from the cells. The longer the MHC monomers needs to dissociate from the cell surface, the better the TCR usually functions. Another way of evaluating TCR functionality is a high throughput screening by using immortalized T cell clones. Müller and colleagues developed a triple parameter TCR signaling reporter cell line based on Jurkat cells and introduced the endogenous TCR knockout cellular platform in their publication last year. With their TPR cell line, transcription factors NF-kappa B, NFAT, and AP1, which are important in T cell activation, can be measured by using fluorescent reporters. To evaluate TCR function, they first co-expressed CD8 in these T cells to stabilize the binding of TCR to peptide-loaded MHCs, and they also knocked out the endogenous TCR of these cells to improve functionality of the transgenic TCR. Here they confirm that CD8 co-expression is needed for stable binding of MHC multimers to 19 different transgenic TCRs. Using peptide titrations, they tested the functionality of the 19 different T cell receptors in the CD8 positive cell line. For this, they incubated the different Jurkat cells with cells that were irradiated and loaded with different peptide concentrations and measured T cell activation by using the NFAT GFP reporter signal. Based on a dose response curve, they calculated the half maximal effective concentration, so the EC50 of NFAT reporter activity. The green dots indicate the best function in TCR. So with this screening, you can actually get an unbiased, easy readout of TCR functionality. After carefully selecting TCRs and successfully expressing them in T cells, the final step would be the clinical application. After isolating and characterizing TCRs, the next step would be to choose the best functioning one or maybe several ones. This TCR then has to be expressed in isolated donor cells, which in the final step can be infused back into the patient, for example, for cancer treatments. With all the information we gave you today, we want to encourage you to learn more about T-cell biology and maybe even go as far as treating patients using the isolated TCRs. We hope that the ideas we proposed today can help you to utilize MHC multimers to do so, or at least give you a broader understanding on how multimers can support you in answering different research questions. We want to take the time and show you how we at IBA Life Sciences might be able to support you in your research efforts by quickly introducing the products that were mentioned in this webinar. For pre-enriching CD8 positive T cells, we offer products that are suitable for affinity chromatographic or magnetic cell isolation. Both methods are based on our traceless affinity cell selection approach, and therefore you will get completely label-free cells after the isolation. The affinity chromatography method is especially suitable for high numbers of target cells, as about 100 million cells can bind in a column. For the next step in the process, so the staining and sorting of antigen-specific T cells, we offer different MHC streptomers. We have, for example, various MHCs that target CMV-specific T cells, but also some that target T cells that recognize tumor antigens. These MHC streps can be flexibly combined with fluorescent streptectin or streptectin conjugated to magnetic microbeads. So not only fluorescent staining and sorting is possible, but also magnetic isolation. For the optional expansion steps after sorting, we offer our CD3/CD28 streptamer kit. The advantage here is that you can terminate the stimulation at any given time simply by adding biotin to the culture. At the same time, you get cells that are completely free of any reagents, because similar to the MHC streptomers, all reagents dissociate from the cells once biotin is added. You can also have a look on our website if you want to learn more about our products. Thank you for joining this webinar. If you have questions or if you have trouble finding the mentioned publications, don't hesitate to contact me. I'm also happy to receive your questions now in the WeChat group.
感谢 Dr. North-Lona 的精彩讲解，给我们分享了 MHC TCR 相互作用、分选方法、应用案例等几个方面。也感谢大家的宝贵时间，相信我们都有所收获。像 North-Lona 博士在讲座中提到的 IBA 的细胞分选扩增平台，以可逆的分选试剂和能获得无标记 label-free 的细胞而著称。主讲座中提到的 MHC streptomer 分离抗原特异性 T 细胞方法之外呢，还提供3种分选解决方案，分别是亲核层析柱、磁珠和荧光分选，满足多种多样的实验需求。根据您细胞数量、不同的样品类型，提供对应的解决方案。IBA 细胞分选方法独具特色与创新，实验中不使用传统的高亲和力抗体，保证温和化的分选流程。选后试剂不残留，助您获得 label-free 无标记的目的细胞。提供的方法能有效提升纯度、得率及细胞活率，受到热切的关注及好评，非常值得推荐。现在，我们的小助手已将大家的提问收集起来了。IBA 的专家稍作休息后，会在微信群里统一解答大家的提问。如果大家还有更多问题，欢迎大家在微信群中继续交流。加入微信群的方法是，请大家注意屏幕下方的交流群栏目，扫描加入微信交流群。关于本次课程的视频回放，请扫码屏幕下方的公众号里面的二维码。回放将在1到2天后放在达优学堂公众号里面，方便大家回看。感谢大家的参与，我们下次再见。